Hello, my name is Lauren Davis, and today I will be discussing recent advancements in structure and functionality of 3D bioprinted cardiac tissue, as this is one of the fastest growing fields in biotechnology, and it has the potential to make an impact on research and clinical practice in the near future. Since the early 20th century, cardiovascular disease, which encompasses a wide range of heart and vascular conditions, has been the leading cause of death in the United States and worldwide. Recently, cutting-edge advancements for applications in device testing, disease modeling, and CVD treatment have been in the field of 3D bioprinting. This additive manufacturing technique utilizes machine coding to layer biomaterials and produce biologically mimetic tissue structures. Knowing its potential to directly impact the over 128 million people living with CVD worldwide, it is imperative to analyze recent advancements in emulative material structure and advanced tissue functionality. The most commonly bioprinted constructs are blood vessels, cardiac patches, heart valves, and whole hearts. The current goal of research in this field is to produce accurate biomimetic models of these structures, both structurally and functionally, for use in research and clinical applications. First, the structure of these constructs must mimic typical cardiovascular tissue. Research in tissue engineering has provided significant data on the natural mechanical properties and microstructure of hearts and vessels, but it has proven somewhat difficult to translate these findings into the 3D bioprinting field, which has left most constructs far too weak in nature. Cardiac tissue, like other soft tissues, is naturally viscoelastic, a challenging characteristic to replicate with most synthetic and biological bioinks. In 2018, it was discovered that carboxyl functionalized carbon nanotubes significantly improve this mechanical property, providing additional networking to the tissue that would leave it compliant yet strong. Another way to increase the strength of 3D bioprinted tissue is to cross-link the material through physical or chemical means. Physical cross-linking typically involves metal ions or electrostatic interactions, while chemical cross-linking most commonly utilizes UV light, thermal heat, or known synthetic chemical crosslinkers. A specific bioink structure challenge is seen when printing vessels. Bioprinting vessels with open lumen that does not collapse or rupture when put through a perfusion system can be difficult to accomplish. Luckily, in 2016, a Seoul University group was able to construct a vessel using a blended bioink technique of GELMA and PEGTA, which also helped the vessel function. Structure of cardiac patches is equally difficult because the tissue must be porous enough to allow for ingrowth of microvasculature. In 2019, a new bioprinting method, freeform reversible embedding of suspended hydrogels, was utilized to print specific structure based on imaging from a real patient's heart, indicating that it is possible to produce realistically structured cardiac patches. While 3D bioprinting structures that can withstand natural mechanical loads is helpful, these structures must also perform functionally similar to the anatomy. To accomplish this, it is necessary to produce prints with various concentrations of cells and other natural components that will encourage tissue adoption, ingrowth, and repair. However, ensuring these cells survive the printing process can be technically challenging. One way to overcome this issue involves the shear stress used when printing the tissue. It has been found that increased shear stress from the nozzle leads to cell death, particularly on the outside of the nozzle, corresponding to a typical shear stress distribution. To improve viability, shear stress in the nozzle of the extrusion tool is decreased, and shear thinning bioinks are used to put less stress on the cells. One notable cardiovascular functional achievement was when tissue constructed with human-derived cardiomyocyte progenitor cells survived when transplanted into a mouse model for 4 weeks. This study not only proved that these constructs are viable in vivo, but further provided motivation to continue enhancing cell viability via stem cells. Specific functions of bioprinted materials are also being explored. In 2020, a one-step method was developed to create bilayer vessel with human umbilical vein endothelial cells on the inside of the lumen and smooth muscle cells on the exterior surface. Having these cellular components organized in a stable bilayer greatly improved vessel performance. Cardiac patches must also perform more unique functions that ideally involve cell alignment, pacing, and integration of vasculature. A new printing method, microscale continuous optical printing using a GELMA bioink, successfully produced well-aligned mouse ventricular cardiomyocytes faster than ever before, which even contracted with significant force. One major issue that will affect bioprinting in the future is that the structural complexity of unique cardiac constructs is incredibly difficult to mimic. This is especially true when scaling up a 3D bioprinted construct for applications like whole organ printing, because advancements have been made in microstructure synthesis, but performing this on a large scale has not yet been achieved. Another complication is with the cells. Malignancy or teratoma formation present potential physical risks, the cost of using cells is very high, and the process of obtaining these cells presents ethical issues. Even when used successfully, most of these cells are newly differentiated, so they cannot perform the same functions that a mature cell does. As the medical device industry expands, it is becoming clear that current methods of testing cannot fully demonstrate reactions and functionality. 3D bioprints have the potential to save essential resources in laboratories by having most test factors in one environment, and they may improve the safety of devices before they are used in animal studies and clinical trials. Living tissue is inherently complex, so having a model to mimic the functions and reactions taking place in real tissue would greatly advance the entire biomedical research process. With bioprinted tissue, it may be easier to induce more accurate disease states and would hopefully allow the environment to naturally develop comorbidities and complications that are missed in other models. The more complex end goal for 3D bioprinting is to use these constructs in the body to replace and repair damaged tissues. Throughout the past decade, vessel and patch constructs have become increasingly successful in testing, and research is beginning to produce various whole heart structures and, separately, functioning Purkinje networks. By combining the fields of additive manufacturing and personalized medicine, 3D bioprinting has the potential to entirely revolutionize biomedical research and clinical treatments for cardiovascular disease. With recent advancements in structure and functionality of constructs, lab-grown transplants and benchtop beating heart models may not be too far in the future.